I welcome you all for another lecture, which is a continuation series from our side to give you a practical insight on various aspects of HPTLC analysis in different fields. Today, I welcome you all on behalf of Anchrom team and hope that this lecture gives you a very insight of the entire aspect of food analysis with HPTLC. With HPTLC, food analysis is highly simplified and easily done with lot many samples in a very short duration. And today I have my colleague, Mr. Ramakant Yadav, who is an application specialist working at our laboratory. He is immensely experienced in the field of food analysis using HPTLC. He has worked with several national food laboratories and also working currently on different projects which are highly challenging in the field of food analysis. And that's why we have deputed him for taking up today's lecture, and he'll give you a good insight of the entire aspect of food analysis and the data obtained with HPTLC, which can be very easy to understand and easily interpreted for different kinds of analytical problems. He has been throughout India at different institutes and industries to give lectures. He has also worked in the pharma industry to give them insights on usage of HPTLC. And with his immense knowledge, I now transfer the session to him, who will give you a lot of insights as previously discussed. So with this note, on behalf of entire Anchrom team, we will start the session. Thank you all. If you have any questions, you do write it at question Q&A box, which is there at the bottom. And for questions which we'll not be able to take during the session, we'll be answering those personally with your emails, uh, registered email addresses which you have sent to us. So with this note, I will now hand over the session to Ramakant. Yeah, Ramakant, you can now take over the session. Thank you all and hope you have a wonderful knowledge session on HPTLC in food analysis data interpretation. Thank you.
Thank you, sir. Thank you, Dr. Sekat, sir. Ankita, my voice is clear?
Yes, Ramakant, your voice is clear.
Okay. So, good morning, all. I, Ramakant Yadav, welcome once again for all the participants in this webinar, which is organized by Anchrom Enterprises Private Limited, India. So, this webinar is specifically for HPTLC analysis in food. How HPTLC is useful, that I will explain in this webinar. So I will be starting with a few slides about TLC, what are the disadvantages of TLC, and how HPTLC is useful to overcome these disadvantages. After that, a few instrumentation. After that, I will show some applications of HPTLC in food analysis. And at the later part, I will show one analysis of food dyes. So I will share my screen. So I will start with the topic. Today's topic is HPTLC instrumentation and interpretation of HPTLC data in food analysis. So we first, what is TLC? TLC is thin layer chromatography. This is a technique by using by which we are going to separate the components from a mixture, and after the separation we are going to identify and quantify. So earlier it was TLC, but now the latest is HPTLC, high performance thin layer chromatography. So Just a comparison between these two techniques. So when you perform any analysis by using thin layer chromatography, there on the right hand side you can see the images. There are less separation. The separation is affected because of many factors: application, mobile phase, saturation, development, humidity. So due to these many factors, the separation is affected, and due to this problem, proper identification is not achieved, and quantification is affected. So all this problem is overcome by using HPTLC. On the left hand side you can see the HPTLC image of ginseng species. So here you can see in TLC only one marker is used, but it is not properly identified and quantified. But in HPTLC we can use multi markers, more than one marker, and we can easily identify in all our sample because of the separation. Main reason why we are getting this separation is because of the type of application. In HPTLC we go for band application. Because of this band application, the separation, the diffusion is very less in the plate. Next, when you go for TLC analysis of any component, any food sample or any herbal sample, it is very difficult to say whether how many components are present. If it is a color sample, so you can identify on the basis of color that whether there are 4 colors or 5 colors. But if I want to go for quantification, it is very difficult because the separation is not there. So you cannot say that pink color is present in this much quantity. So to overcome this problem, you can use HPTLC, in which you have spot application and band application. And in both the application pattern, you will get the best separation and best resolution. So in this slide, you can see there is a comparison of two stationary phase. Previously, uh, before the introducing the HPTLC plate, it was TLC plate is used. TLC plate 20 by 20, which is you can see on the left side. So by using 20 by 20 plate, uh, we are going to develop the plate in 15 centimeter, so which require more solvent and more time is required for the development. But when HPTLC plate is used, in half of the plate your same sample is getting separated within less time and with less solvent. So when you use HPTLC, you are doing the miniaturization with respect to solvent, less time and more samples. So this is the advantage of HPTLC stationary phases. TLC versus HPTLC. Both the techniques are same, planar chromatography. But main thing, what is the target? In TLC, you will get rapid results, preliminary results only. You will not get reliable results. Reliable results, reproducible results, you will get it in only HPTLC. As per the cost of technique, TLC, the cost is very low, directly depending on the results you are getting. But when you go for HPTLC, the cost is medium to high as compared to TLC, and low per sample as compared to other technique like HPLC. So you can prefer HPTLC for reliable data, and the cost is also very low. So why HPTLC? Because it is simple, reliable, and fast technique. In HPTLC, there is no contamination of chromatograph, because for each and every time we are going to use new stationary phase for new sample analysis. It is simple because the software is very simple, easy to handle. The process, the practice of HPTLC is very simple. Low running cost and maintenance. The running cost is very low because the solvent consumption is very less. The stationary phase cost is very less, and the maintenance of HPTLC instruments, it's very less. And main thing, double confirmation results. You are going to get, you can check the result by your eyes, you can get the data, electronic data, and you can go for bioactivity studies also. From a single plate, you will get a large data, that is same sample image in UV light, visible light, fluorescence light before derivatization. And after the development, you can go for the scanning, single wavelength scanning, multi-wavelength scanning, and spectrum data. And after this, you can go for the derivatization of the same plate. So because of this derivatization, the data is going to be changed, and that change data is an additional information for the same sample. So for the same single sample in the single run, you have lot of information which you can use it for further analysis. Practice of HPTLC. First is the software activation. Before software activation, sample preparation is one of the major uh, steps. You can follow the sample preparation as per the various matrices, whether it may be food, it may be herbal, or any type of sample. After that, you will fill the parameter in the software. After that, you will go for the first step, that is application of standard and sample onto plate. Next step is the development, that is the separation of components from a mixture, which will be done by using a stationary phase and a mobile phase in a closed chamber. After the development, next step is image documentation. You need to capture the plate in three different illumination: UV, visible, and fluorescence light. Next step is the densitometric scanning. Depending on the components, you can go for single wavelength scanning, multi wavelength scanning, and spectra. After the scanning is over, your data, this is a non-destructive technique, so the component will remain on the stationary phase, so that you can use it for further study, that is bioactivity, effect directed analysis. You can take that particular separated analyte to MS, you can take it to IR, NMR also, and if required, you can go for derivatization. And if derivatization is performed, you need to go for again photo documentation and scanning. This is the sample preparation as per USP chapter 203. You can use this preparation for those food material which is having the botanical origin. So for that type of sample, you can dry the sample, powder it, and use this process. This process says 1 gram of sample dissolve in 10 ml of desired solvent. After that, sonication for 10 minutes. After that, centrifugation for 10 minutes, and you can take the clear supernatant for your analysis. So you can see how the sample preparation is very simple, and it will take very less time, and no need to prepare lot of uh, tedious process is not involved in HPTLC sample preparation. So complete system of for HPTLC. Here you can see first module is the applicator, next is the development module. After that, the both two modules are used for derivatization purpose. So this is only for the application working per means application development of the stationary phase. But after that, the fourth module is the scanner. It is for the chromatographic evaluation. From the scanner, you will get the corresponding peak profile depending on the concentration of analyte present on the plate. Next is a visualizer 2. Visualizer 2 will give you the plate images in three different light, similarly as shown in the image. Next is the interface 2. Interface 2 is the hyphenation which is added to this HPTLC technique. By using this interface 2, you can take the analyte from the stationary phase to other technique, where you can confirm the you can get more confirmation, and you can go for further characterization of that particular analyte. As per the regulatory acceptance of HPTLC, it is official in USP, USP official from 2018. Yeah, European Pharmacopoeia, USP chapter 621 for scanning densitometrically for quantification work, USP chapter 202 for identification of fixed oil, and you have USP chapter 1064, that is that will say about the parameter which you are going to use it for the analysis. It is official in IP, Chinese Pharmacopoeia, and WHO guidelines for herbal medicines. One by one, I will go for the each and every module. So for the sample application, we have two module. One is Linomet 5, one is auto sampler ATS4. So depending on the number of sample, you can select from one of these module. For more number of sample, you can go for ATS4. For less number of sample per day, you can go for Linomet 5. Both the applicator require a gas. Both the applicator can give you spot application, band application in a very precise and sharp mode. Both is software control, so it will meet the 21 CFR regulatory compliance. So it is GLP compliance also. It is IQ OK compliance. So these are the following type of application which you can get it in the by using this applicator. First one is the normal quantitative application. This type of application you can use it for qualitative as well as quantitative. Next type is in situ cleanup. This is used for those type of sample which is having some oily matrices, fatty matrices. So you can apply the sample containing that, and you can remove these matrices on plate. A third one is the micro preparative technique. So if you want to isolate the component, so you can apply the sample and standard into a higher quantity, and the more you load, the more you can isolate. Last one is the superimpose technique. By using this applicator, you can over spot the standard and sample on the same band. That will be used for verifying the result, cross checking the results whether it is present or not. So it may be used as an internal standard also. So these are the type of application which you can perform by using the applicators. Next is the development module. So for development, we have twin trough chamber, which consists of three different dimensions: 10 by 10, 20 by 10, and 20 by 20. Next is the ADC2 automatic developing chamber. So in ADC2, many parameters will be controlled like saturation, humidity, and automatic development, automatic drying of the plate. So no manual thing is involved. So for getting reproducible results, you can use ADC2. Both these two techniques, TTC and ADC2, is isocratic separation. You can perform the separation in uh, one step, but for multiple development, you can go for the AMD2, which is present on the left side of the screen. So in this module, you can develop the same plate into number of steps, like first step with different mobile phase, and in the second step you can change the composition of the mobile phase. You can change the polarity of the mobile phase. So if the work is required in this sense, so you can use AMD2. Next is the photo documentation, that is visualizer 2. So here you can see on the top you have this industrial grade camera which is capable of taking a high HDRI mode image in HDRI mode. So in why HDRI mode is required for, that is required for fluorescence image. So by using this visualizer, you can get high good quality images in dark condition, and you can capture the image in 254 and visible light. There is a slit to observe the plate under this light, and it is also software control. So directly you need to give the command through software, and it will capture the images automatically, and this image will be stored for lifetime. Whenever you want, you can use this data for further analysis. uh, from the visual plate image, you can get the uh, peak profile, uh, and you can get the single means you can, if the image is taken in three different illumination, so you can get the three different illumination peak profile simultaneously also. You can go for semi quantitative work by using this TLC visualizer tool. Now next is the HPTLC uh, scanner 4. So the scanner is the heart of HPTLC. In this you have the range of 190 to 900 nanometer, which will cover your UV visible and fluorescence range. So after the development, whatever component is present on your stationary phase, you can easily go for the scanning of that component. From the scanner, you will get the corresponding peak profile depending on the concentration of that analyte present on plate. Uh, and you can go for its quantification by single level, multi-level. You can take the spectra like this. and you can check whether the spectra of pure standard is matching the spectra of standard present in your sample. So after checking the spectra, you can confirm on its identity. So in this scanner, it is fully automatic scan plus spectra, and you have the range from nanogram to picogram depending on the component your main marker. If it is in if it is fluorescence active, so you can go beyond this limit also. After the spectrum, you can go for the linear curve and you can get the quantification. So this is the type of spectrum analysis. You need to match the spectra for confirmation of any particular drug in your sample, in your food matrices also. If the overlapping is similar to this, then you can confirm the presence. If the spectra is not overlapping with each other, so it is not present. This is the linearity and calibration curve. So this is also possible by using the scanner 4. Next is the derivation modules. So we have two different modules for derivation. One is immersion device 3, which is used for dipping the plate, stationary phase plate, in the derivatizing reagent. You can dip 20 by 10, 10 by 10, and 20 by 20 plates. So one is the dipping technique, another one is the spraying technique. So this is the automatic derivatizer, which uses a piezoelectric head, which will convert the droplets, bigger droplet, into fine mist, and that fine mist will be sprayed onto the plate. So in this hood, the spraying of derivatizing reagent is done automatically, and after the spraying is done, automatically the uh, hazardous fumes will be aspirated into the bag bottle which is present, and it will ensure that analyst will not inhale any hazardous fumes. And major thing of this module is it require very less volume of reagent. 3 ml of derivatizing reagent is used for 20 by 10 plate, and 6 ml for 20 by 20. Where in the previous immersion device you require 150 to 200 ml of the reagent. So both the techniques is good. Last part is the TLCMS interface too. So here you can see like this, we need to keep the plate on below the illusion head, and after that this illusion head will elute that particular separated component, and it will transfer it to MS, wherever where you will get the mass data, and you can take that particular analyte to IR also. So like this, you need to make the connection. The inlet of interface will be connected to the HPLC. If you have HPLC, so you can connect directly, or if you don't have, so you need to have a HPLC pump. So the pump will be connected to the inlet of the interface, and the outlet will be connected to the MS. So directly within fraction of 10 to 20 seconds, you will the analyte from the stationary phase will be dissolved in the carrier solvent, and it will be carried through the MS, and in between there is a frit which will prevent that silica gel to pass. So there is no harm, means the silica will not interfere in the MS part. So now the application of HPTLC. So HPTLC is widely used in various fields like herbal medicine, food and feed, pharmaceutical application, industrial application, forensic, cosmetic, and many more. So in herbal medicines, it can be used to identify various markers, depend pharmacological active marker you can identify and quantify. After that, in food and feed, it can be used to identify the adulterant. It can be used to identify the nutrients. You can go for the um, fingerprint work. After that, in the forensic, it can be used to identify uh, psychotropic drug, some drug of abuse, you can identify, you can go for pesticide analysis also. And in pharmaceutical application, you can go for the impurity profiling. Majorly it is used for this work, and identification and quantification of APIs in the finished drug. So food and botanical sample analysis on HPTLC. So various type of analysis can be performed by using HPTLC for foods. First is the fingerprint analysis for food. Next is the identification, quantification, antioxidant, and class of compound analysis. So what is HPTLC fingerprint? So fingerprint is the electronic image of the visual HPTLC chromatogram. So what do you mean by this? So for example, if I have any if I have kurkuma, haldi sample, so I will dissolve in a particular solvent and I will apply that particular onto the stationary phase, and I will develop in a mobile phase. And after the development, the particular haldi sample uh, will have some the separation pattern like this. The separation pattern will be I can take the image in three different lights, so three different separation pattern I will get. So this separation pattern is called the HPTLC fingerprint. And how to evaluate this fingerprint? So here you can see in the third track, there are different bands of different colors. So it may be of different components present in that particular sample. So depending on the color, intensity, and RF of these particular bands, you can go for the quality check. You can go for the pass fail test also. So here you can see, with the help of HPTLC fingerprint, you can identify various species of piper. Piper, you can see Piper nigrum, Piper longum, Piper rectifectum. So the fingerprint is different. Due to change in the species, the fingerprint is changing. So here you can see Piper cubeba, it is having a very prominent orange color band here. So that is not available in any of the fingerprint pattern of its species. So this particular band can be used as a specific marker for this Piper cubeba. If any adulteration of other species is done in this Piper cubeba, you can easily identify. Next, here you can see fingerprint of fats. Means in food, this is the application of foods. So here you can see various fats. We have done the fingerprints of that. Here you can see first three is the butter. Next is the sunflower, cow ghee, pure ghee, like this. So we can get the fingerprint pattern, and with the help of fingerprint pattern, you can check whether the oil is added in butter, or it is added in cow ghee, or whether two oils are mixed. So this is the main advantage of fingerprint. Simultaneously, you can develop many number of samples. Here you can see 25 samples we have applied on the same plate, and we have developed the fingerprint, and we can go for the comparison. We can compare the fingerprint profile of butter with sunflower with cow ghee. We can compare the fingerprint pattern with various market butters. Comparison of multiple samples. So here you can see in this slide the comparison of various species of curcuma is done. So curcuma longa, curcuma xanthorrhiza, there are various species of curcuma. This comparison is not from the same plate, from different plates, but the parameters are fixed. By fixed parameter, the tracks from different plates is taken, and simultaneously the comparison you can see. So in all the samples, you can see this particular red color band, dark red color band in the center zone is prominent. It is present in all the species, but due to few of the particular phytoconstituent, the fingerprint will change. So here you can check that also. Next, one of the best example of this fingerprint, by using this fingerprint, you can say whether curcuma aromatica is added to curcuma longa or curcuma longa is added to curcuma aromatica. So how I will explain. So here you can see in the first track when it is 100% curcuma longa, so there is no violet color band present here. But when you add 10% of curcuma aromatica, the presence of this violet color band will show here. So as you add more and more amount of curcuma aromatica, the intensity, the on plate concentration will increase, and you can go for the quantification of these also. And when you have 100% curcuma aromatica, there is absence of this particular bottom band, that is this methoxycurcumin. So this band and the upper purple color band can be used as a specific marker for specific species. This they can be used to identify the adulteration of these two species with each other. HPTLC identification analysis in food. Here, identification of monosaccharide. So various sugars, for example, fructose, maltose, all the monosaccharides can be separated in the same plates. And after the method is developed, uh, in the next slide, what I have done, I have sorted that with respect to their RF value. I have sorted that standards, and in the previous slide, it required 11 tracks, but here the standards are uh, fixed in 5 tracks only. And here you can see in the extract samples, I can easily identify this sucrose. And after the identification is over, you can go for the quantification. So this identification of monosaccharide you can perform by using any sample. It may be a sweet sample, it may be a juice, it may be any nutraceutical supplements also. Next is FSSI permitted 9 synthetic food colors. So here you can see all 9 permitted colors are separated in the same plates. So after setting the method, these colors, synthetic colors, were identified in various food matrices. For example, first you can see kala khatta. Kala khatta is the juice which everyone's know. After that, mukwas. So in mukwas you can see tartrazine is present, brilliant blue is present, Allura red is present. After that, erythrocin is present. So next sample is candies. Candies contains more colors. Halwa burfi, you can identify these two colors from halwa burfi. Jalebi, laddu, fruit juice, kulfi. So after the method is set, so you can use this method for any food articles, whether um, all the food matrices is acceptable. By using this method, you can identify, you can go for the quantification. As per the FSSI limit, 100 ppm is only allowed for synthetic permitted food color. So we can set a limit test analysis for this work. HPTLC quantification analysis in foods. So here you can see quantification of synthetic food color, sunset yellow and tartrazine in T sample. As per the FSSI manual, T sample, the raw tea powder should not contain synthetic permitted color. But if it is detected, so you can go for the quantification. So by using multi-level calibration, we have quantified that particular tartrazine and sunset yellow in the green tea powder. After that, quantification of curcuminoids from curcuma longa root sample. Uh, Curcuminoids is a mixture of 3 different curcumin, that is curcumin, bismethoxycurcumin, and this bismethoxycurcumin. So all the 3 curcuminoids can be identified and simultaneously quantified. It is very easy to quantify the more than one marker, more than one component in any of the samples. Quantification of caffeine in tea powder. Not only tea powder, you can go for the quantification in coffee and soft drink, energy drinks. So you can go for the estimation of this caffeine. you can go for the class of compounds. So if you want to check whether the my food component is having uh, which class of compound, for example phenolic, tannins, polyphenols, or saccharides. So you can check this various class of components by using HPTLC, uh, whether it contains steroids, saponins. So all you can perform by using HPTLC. Now, further application of HPTLC by you can go for the detection of papaya seeds in black pepper sample. So nowadays these black peppers are adulterated with dried papaya seeds. So we have developed the method for this, for detecting this. So here you can see on track number 2 there is a particular band present at 0.24 RF. So this band can is only is a specific for papaya seeds. When you detect this particular band in black pepper, so that means it is adulterated with this papaya seed. So you can, after the identification, you can go for the quantification. Detection of Sudan dyes in chili powder. In this slide, there is only 4 Sudan dyes, but you can go for all 9 class of Sudan dyes, and you can go for the detection and quantification in chili powder or any food articles, like sweets, like any spices, because it is added in spices to increase the uh, uh, quality. It is mostly added in chili because to give more redness, and because of that the profit will also increase. So it is non-permitted dyes, it is a carcinogenic dye, so it should be absent, but if it is detected, so you can easily identify and quantify. Next, quantification, identification and quantification of melamine in milk by HPTLC. So this is a major problem, because due to if the milk is pure, the the protein content is proper. But when the milk is diluted to gain more profit, the milk is diluted, and with respect to this dilution, the amount of protein content is reduced. So to make that protein content in the permissible range, they add melamines. The melamine is a 5 amino components. Due to this, the protein content is proper. So but it is very harmful for human health. So you can detect this melamine and you can go for the quantification. And it is mostly added in milk only. Detection of sibutramine in slimming product by HPTLC and confirmation by HPTLC-MS. So sibutramine is an drug which is it is not allowed, but it is added in slimming product like green tea, whatever are available. So they are adding the sibutramine, oristatal, so to reduce fat. But this is very harmful, so you can go for the detection. And here you can see after the this drug is identified on the stationary phase, we have performed the MS part. The illusion of that particular component from the sample is done, and you have uh, got this M by Z value also. And you can go for this type of confirmation. You can go for the identification of lactose in food and beverages. So it is very lactose and sorry, determination of sucralose and fructose in food and beverages. So uh, in food and beverages they are adding these sugars, so to increase the sweetness. So you can identify, you can go for the quantitative work in various samples, and there is a limit for these food and beverages. So you can go for this fructose limit and sucralose limit test analysis. Adulteration of toxic argemone oil in mustard oil by HPTLC. Mostly the mustard oil, on if you purchase any mustard oil packet or any bottle of mustard oil, it is written it is free from argemone oil. So argemone oil, it is most of the time it may be a contamination or it may be an adulteration. So due to this argemone oil, there is a presence of two alkaloids, sanguinarine and dihydrosanguinarine. So these two alkaloids are not good for human health, and it should be absent. But if it is present in the mustard oil, you will get these two prominent band in the sample. So we have developed this method in-house Anchrom lab. So it is very simple and fast method, and it is having the fluorescence activity. So you can go for the quantification in very low level also. Limit test for sorbic acid in cheese. So sorbic acid, it is an preservative which is added in cheese. So this is allowed preservative, but it is having the limit. It is having the limit of 1000 ppm. So if the sorbic acid is added in more quantity, so you can detect this by using the limit test option of vision cat software by HPTLC. So it will be very simple analysis. You can just set the 1000 ppm limit, and if the sample is containing sorbic acid below the limit, it will automatically pass, and above the limit it will automatically fail. Detection of nicotine in tobacco, gutka, pan masala, and mawa sample. So nicotine, it is an alkaloid, it is an addictive drug. So as per the FSSI manual, nicotine should be used as raw material, raw. It cannot be added in any of the food like pan masala, uh, mawa samples. So detection of this nicotine in this sample is not to be present. So you can identify this, and you can nicotine is one of the good drug which is which doesn't require derivation. After development only you can identify, and you can take the spectra, and after taking the spectra you can confirm the presence of nicotine, and you can confirm more by going for the derivation. And after derivation you can see nicotine is showing reddish brown band. Okay. uh, by using HPTLC fingerprint, you can differentiate between various species of uh, various source of honey. So here you can see, for example, if the honey is from raspberry source, if the honey is from clove, it is different. Due to if the honey is going to change, if the source is changed. So various type of honey will have different type of fingerprint pattern, and with respect to this pattern, you can say whether it is from the raspberry honey, or it is a clover honey, or it is other species of honey. Detection of BHA and BHT in ghee. So here you can see these are the antioxidants which are added in ghee sample. So there are various already methods are available, but the methods are very tedious. And by using this HPTLC method, you can identify and quantify this antioxidant in very less time span. So here you can see we have in ghee sample it was not detected, it was very good, but when we spike, so the by using this method it is very prominently it is getting separated, and you can identify that. You can go for the vitamin C identification in fruit juice, you can go for the quantification of it, you can go for the quantitative determination of various amino acids. You can see leucine, valine, all the amino acids you can identify it in food samples. uh, you can identify in nutraceutical supplements for example bodybuilding supplements. There are many supplements which claim more amino acids, so you can go for that type of sample analysis also, and you can check whether the pure amino acids are added or not. Determination of lactose in milk products product and lactose free milk product also. So this can be used to detect uh, if the product is claiming it is lactose free, so you can check by using this method whether the lactose is present, and if present, so how much it present. Quantification of 5-hydroxy methyl furfural in honey. So this 5-hydroxy methyl furfural is an indicator for the freshness of honey. If the honey is going to be old, the amount of this marker going to be increased. So depending on the quantity of this marker, you can say whether the honey is pure, fresh, or it is old honey. So identification of preservative in tomato sauce, you can see methyl paraben, propyl paraben, sodium benzoate. There are various preservatives which are added in tomato sauce, other food products also. So you can easily identify these preservatives. Quantification of piperine in piper nigrum. It is one of the example of similar type of application. Fast determination of benzoic acid, one more preservatives. Quantitative determination of stevial glycoside. This is very good. Nowadays you must be knowing the alternative of sugar. Alternative of sugar is stevia. Stevia is one of the plant which is having various glycosides. This is the replacement of sugar, and nowadays many products are coming by in which they are using stevia as a alternative for sugar. It can be used for bakery product. It can be used for uh, as a in food preparation. It can be used in sweet preparation. So there are many glycosides present in stevia, but few of the glycosides like ribidoside A and B, they are having some means um, pungent pungent taste. So you can identify that particular glycoside, and you can remove this particular glycoside, and you can go for the further processing. So it is very useful for stevia manufacturer, processing company. Total polar components in frying oil. So now by using this application, you can check any whether the food oil which you are using, it is useful, safe after particular time, or it is unsafe. As per the FSSI claim, the polar component should be 25%, less than, not more than 25%, and the triglyceride should be 75%. This is the limit. If the limit of polar component increases from 25% and the triglyceride reduces from 75%, the oil should not be used. So here you can see at zero point 0.2 RF in the sample, this is the band of triglyceride, and all the track number from 9 onwards, it is various oil uh, after heating at 1 hour, 2 hour, 3 hour, like this, at different intervals. So after checking at 4 hours, the triglyceride is getting decreased, and if the triglyceride is decreasing, then alternatively the polar component will increase, and that will lead to change the quality of oil. Why this is happening? Because when we use any food article for frying, it will contain some water. For example, potato, it already contains some water in it, and due to open cooking, there is air oxidation taking place, then there is formation of free radical, and that will lead to change the thickness of oil. If you know, if you just remember, after heating of oil for 4 hours, 3 hours, the oil viscosity is changed. So the viscosity is changed due to this reason only. So you can check whether the oil is good for health after 4 hours of heating, or it is not. Detection of palm oil adulteration in edible oils. So here you can see palm oil as a, it is not harmful, but due to its cost, it is of very low cost. So it is added in other high cost edible oils. So you can detect the adulteration of palm oil in other edible oil by using HPTLC. So palm oil has some distinctive bands, one, these two bands, and at this band. So when palm oil is added to any particular mustard oil or sunflower oil. So these three bands can be used to detect the presence of palm oil. Fingerprinting profile of butter and margarine. So the major adulteration of butter is done by using margarine, that is white butter. It is nothing but uh, a mixture of vanaspati ghee, which contains already palm oil. So here you can see how I can differentiate pure butter and margarine. So here you can see track number 23234. It is pure butter. Uh, Two are from market sample and one is homemade butter. So if the butter is pure, they will have the similar fingerprint pattern. And if here you can see track number 656, these are the real life confiscated butter from FDS, but they are the fake butter because they are not having the similar fingerprint pattern. So on this basis, you can differentiate pure butter or adulterated butter. Detection of nicotine herbal hookah. Recently, a case in High Court was there. Many samples of herbal hookah was confiscated for checking the nicotine. So we have developed the method for this, but it was good that nicotine was not detected in any herbal hookah. Identification and quantification of monosodium glutamate in 7 food products like noodle pasta. So as per the FSSAI claim, you can add MSG, monosodium glutamate, or also known as Ajinomoto. This is a taste enhancer. This is added in various Chinese product, Manchurian, with various products are using this, but this should be added in a limited quantity. It can be used as a seasoning, but in a limited. So what they are, many preparing companies or particular individuals, they are adding this MSG in the noodles in raw material only. It may be added in noodles, it may be added in pasta, but this practice is not allowed as per the FSSI. This MSG should be added only in the masala. Masala means that spices packet only. It should not be detected in noodle pasta. So you can detect and quantify this. Quantif Quantification of glycerol in tobacco. Why glycerol is added? By using this glycerol, it is an humidifying agent. It will increase the palatability, shiny, means it will enhance the burning time of the tobacco leaves. If the leaves are dried, so it will burn in very less span. But if the leaves are somewhat humid, containing more moisture, so it will require more time. So many cigarette companies, they are adding or may not be adding this glycerol in the tobacco. HPTLC, UVM, and MS of caffeine in energy drinks. So caffeine can be used in energy drink, but up to a particular limits. So you can go for the identification of caffeine or similar uh, addictive drugs in the energy drinks, and you can go for the MS work. Here you can see after the identification in the first plate image, we have performed the MS part. So after MS, the particular zone is eluted, and we have checked the M by Z ratio, and it was similar. Fingerprint profile of fixed oil. So you can get generate the fingerprint also for various type of oil: corn oil, peanut oil. uh, so each and every oil will have different fingerprint pattern, and if two oils are mixed to each other, so the fingerprint pattern will vary, and from this you can identify whether the oil is pure or mixed. Determination of aflatoxin B1, G1, B2, and G2. So aflatoxin, the carcinogenic components, which is not uh, added, it is if the proper storage of food grains is not done, if the proper storage of feed sample, animal feed is not done properly, so there may be a formation of fungus, and due to this aflatoxin is going to be interfere. So you can easily identify these all four aflatoxin, and you can go for aflatoxin M1 also, not only B1, B2, G2. You can go for M1 also in milk. Identification of nimesulide and diclofenac in Ayurvedic tablets. So here you can see these are the drug which is used as a pain relieving drug. So if it is added in Ayurvedic drug, so it is very wrong thing. It is not allowed. So why they are adding in Ayurvedic formulation? Because to increase the effectiveness of that Ayurvedic tablet, powder or syrup, they are adding these APIs. These are the APIs. Due to this API, the effect will be fast. So here you can see on the first track it is pure nimesulide API, second track diclofenac, and this is a mixture of standard. And in the tablet I have uh, uh, I am getting the same band and the same RF value. And after the visual confirmation, I have taken the spectra, and it is overlapping in the spectra for diclofenac and for nimesulide also. So I can detect and quantify this and uh, stop this adulteration also. Berberin identification in Darwaldi. These are the application for uh, uh, those samples which is having the botanical source. This type of application can be used in nutraceuticals. So here you can see berberin is identified in Daru Haldi. So these are the two different vendors. So if any company which is working with this production, so they will purchase the raw material for a various vendor, and to check whether which vendor is giving proper quality of raw material, you can do this fingerprint. So first two track is of different vendor, last two track is of different vendor. So in between I have used this marker, and you can identify in the first image that last two track of vendor number 2 is having more quantity of that marker. So on this type criteria you can select the proper vendors. Berberin in Berberis aristata, gallic acid in amla. Not only in amla, you can go in any citrus fruit. So you can go for the identification and quantification. Eugenol, uracilic acid identification in tulsi. So here you can see in the first image, eugenol marker can be can be seen and identified, but uracilic acid is not detecting. So this type of component require derivatization. So I have derivatized the plate with ASR, anisaldehyde sulfuric acid reagent, and I have observed the plate into white light and fluorescence light. So after derivatization, you can see the particular band is showing ashwagandha. Everyone must be knowing about this ashwagandha. So you can go for the detection of vithanone A, vithanolide B, vithaferin A. You can use more than this marker also. So simultaneously identification of multiple marker, and you can go for the quantification also. Whether if you go for any Ayurvedic bandar, what you are purchasing that powder, ashwagandha powder, whether the powder is pure or it may be a mixture or adulterated with any other powder. So you can check with this type of work. So these are some literature available for HPTLC, that is ICMR books. There are for 17 volumes. So here you can see the name quality standard of Indian medicinal plants. So most of the medicinal plants are covered here. API for Ayurvedic plants, high performance thin layer chromatography in food analysis by Prem Kumar Jaiswal sir. So many of the food samples were covered here. So uh, these are the following links by which you can connect to us much better, and you can get daily updates regarding the upcoming work in HPTLC, which we are performing in our lab. So you have LinkedIn link, Facebook link, you have the YouTube where we upload daily, means weekly videos of for the latest trends. uh, Anchrom at your service means we are what facilities we are providing for our customers, and not only for a customer, for academics, for students, for teachers. We are providing free training, free analysis, for subsidized analysis. We are providing PhD on and learn scheme also. We are having various MOUs with various prestigious institutes, so they can uh, get the sample analyzed by us. We are going to help in all the field of HPTLC requirements. So we are dedicated to this TLC HPTLC since 1978. So we are the master of HPTLC. Any sample from any field we can perform in our lab, and we can give the proper results to you. Now, I will use 10 minutes. I will use some of the uh, analysis I will show for the food dyes. So I will switch to my colleague PC, and I will show there. Hello, hello, Ankita. Hello. So I will share my screen. So here you can see I have performed few of the analysis of food dyes from various food matrices, so that I have sorted. So I will be showing that. So first is the identification. So here you can see all the 9 permitted dyes are separated in the same plate. So after the identification method is set, what we have done, we have uh, gone for the identification from food samples. So here you can see. So similarly, you will get the plate images from the visualizer. So how to evaluate this data? So this is the raw plate without any RF tool. So you can add this RF tool that will add the RF scale from the application position to the solvent front position. You can get these labels. After that, for checking the low intense band, you can go for the spot amplification by using this. So you can detect the low intense band also. You can change the background and you can go for the uh, various evaluation for resetting. There is also option. So similarly, you have the option to evaluate the plate images in white light, uh, in 254 light, in 366 light also. So this is the image data evaluation. And now I will show here. You can see after the identification is over, I have taken the spectrum here. So I will show once again proper. See, after the in this plate, you can easily identify the particular dyes in the samples. So the RF is, you can check the RF 0.32, and in the sample also it is 0.33, 0.32. Like this, you can evaluate the image data. After the RF is going to match, you will take the spectra. So I have taken the spectra of Allura red in the standard, and Allura red from the candy sample. So after RF is matching, you need to take the spectra. So I have taken the spectra. I will show the spectra pattern. Here you can see the spectra pattern is matching. There is a pure overlapping of the spectra of standard and a band which is detected in the sample. So after matching the spectra only, you can confirm the presence of that particular dye. And after the dyes are identified, you can go for the quantification as I have shown in the data. But as per the FSSI, there is a limit that is 100 ppm for each dye. So here I will show in another plate the how I perform the limit test. So in the limit test, if 100 ppm is the limit, so you need to apply the 100% on plate concentration for each and every standard, and with respect to the sample also the concentration should match. And after that, in the evaluation, we have the option of limit test. Here you can see we have the option of limit test. In the limit test option, you can fill you can select the reference track for example, track number 1 is containing your tartrazine, that is 100 ppm on plate concentration. So this will be your reference, and if in your sample it is present below this, so it will pass, or if it is above that, it will fail. So I have selected the limit test criteria below, and now you can see in the results so here you can see only track number 6 is having that erythrocene, and it is passing because the maximum limit is 2.5 microgram. Here it is 701 nanogram. Next you can see in the evaluation 2, I have done the limit test for tartrazine. So tartrazine is detected in various food matrices in track number 6, 7, in other track also. So wherever it is detected, it will check the quantity, and if the quantity is below the range, it will show this green color bar and it will pass. If it is above the range, so here you can see 2.5, the yellow color line is the limit, and that red color line is the allowed range. If I give 10% deviation, so about 2.5 10%, and below 2.5 it will set as the deviation range. So another example is for sunset yellow. So here you can see all the samples are in the range, so you can use this limit test option, or if you want to go for multi-level quantification, that is also possible. And uh, that's all from my side. uh, Now we will go for the question answer. Vishwajit, hello.
Hello, am I audible, Ramakant?
Yes, yes, Vishwajit, you are audible.
Yeah.
So
So there are questions like analysis of essential oils and volatile oils by using HPTLC. Can we perform that analysis by using
uh,
HPTLC?
essential oils we can perform, but not for the means. Mostly volatile is not properly suitable for this, but you can perform for other oils like non-volatile oils. Volatile oils, because it is an open technique, so you can go for the qualitative work but not for proper quantitative work.
Okay. Thank you, Ramakant. There is one other question like analysis of syrups and removal of matrix interference like sugars and all. Can we do that?
Yes, yes, yes. Syrups will contain what is the criteria, means what is the aim in syrup, what you want to identify, whether the you want to identify any APIs or any food dyes. So it depends on that. We can follow the extraction procedure, and yes, it is very easy to identify and quantify any of the component from syrup sample. Next question.
Okay, thank you, Ramakant. And there are many questions. All the participants ask the questions in chat box as well as in Q&A box. So some participants require methods for different analysis. We will send on their registered email ID. Uh, Ramakant, there is a question about detection. So how many detector modes are available in HPTLC, and whether hyphenation is required always for in detail detection or HPTLC detectors will work?
Uh, in HPTLC, the data are available. It will start. You will get the data from visualizer only. From first, you will get the image data in different lights. For example, UV light, 360 fluorescence light, and visible light. And from this visual data, you can generate the corresponding profile. So this is the image profile. So this profile is only available in three different specific light, and after that you have the scanner is having the range from 190 to 900, so you can go for multi wavelength scanning also. After that you can take the spectra, and as per the requirement of MS. It depends on, for example, if the uh, particular identification is performed by using the plate image. On the plate you can see the RF and the color of that particular standard. So that is one confirmation. After that you can confirm with the help of spectrum, and if you want to confirm on the basis of its mass, so you can go for the MS. If you add MS, so it will be a better confirmative technique. You can go for IR also for functional group confirmation.
Okay, thank you, Ramakant. We will take one more last question, I think.
Yes, yes.
How quantification can be carried out after chromatogram development?
After chromatogram development means it is a separation. In this process, the applied stationary phase with our standard and sample is kept in a chamber containing a mobile phase. After keeping the plate in that, the mobile phase will run on the stationary phase. With the help of capillary action, we will run the plate up to a particular solvent front, and after that we'll remove and dried, and after the dried plate we can keep in the scanner, and we can go for the scanning and quantification.
Okay, thank you, Ramakant. I think there are many questions. We will send the answers of remaining questions on respective registered email IDs.
Yes.
That's it from my side from Q&A. Thank you.
Okay. Thank you, Vishwajit.
Thank you, Ramakant, for such a wonderful session. So participants, I hope the session was wonderful and insightful for you. So we would like to inform you that we will be conducting more such topics on HPTLC. If you are interested that you want to know any such topics which should be covered in detail, you can always email us at lab@anchrom.in. We regularly post content on how to troubleshoot your HPTLC modules, instruments. If you're stuck anywhere in your analysis, you can always refer our website, YouTube channel, or LinkedIn. Please follow us over there. And I would like to inform you that we will be giving the certificates through email 2 weeks after today. So I request you to register for the upcoming webinars also. Yes, and if you have any further queries, we would be happy to take, or else we can end the session, and you can email us at lab@anchrom.in. Hello everyone. The polls were the feedback actually. So yes, we will send you the certificates 2 weeks after today. We'll sort out and send you. The certificates are not e-generated.